Hey everyone, welcome to my molecular biochemistry series, and in this episode we are focused on protein structure. Now there are 4 different levels to protein structure. At the base you have the primary, then you can move up to secondary, and then there's tertiary, and finally quaternary. And that will be the emphasis of this video. At the end I'll also touch on a specific example of a protein in action. Now, in previous videos, I talked quite a bit about the structure of DNA and how it facilitates the formation and production of proteins. But now, since we've already done that with transcription, translation, DNA replication, etc., we want to see what do these proteins actually look like when they are formed. And so first, we'll look at the primary structure, and we can see that it begins with the N terminus and ends with the C terminus. And throughout this sequencing, this polypeptide, you have an amino acid, then a carbonyl, then the peptide bond, then a nitrogen, and then this repeats over and over and over again. Those are how amino acids are linked together via translation, via a peptide bond. And this is done through a condensation reaction. So this is repeated over and over again to form the primary structure. Now, if we move up to the secondary structure, we are going to form our alpha helices and beta plated sheets. Now, the alpha helix is formed via hydrogen bonds, causing a looping action to take place. And you have to imagine this really in 3-dimensional structure, which is not what is drawn here, but this is in 3 dimensions. So you form hydrogen bonds between the carbonyl and the nitrogen in the peptide bond. And this is done roughly 4 amino acids apart. And so this happens over and over again, and the alpha helix forms. Alternatively, you can have a beta plated sheet, and in this case, the hydrogen bond is still the primary force, actually the only force leading to the stabilization of secondary structure. And so you have various different hydrogen bonds here, and it causes a beta plated sheet to form. One other interesting point is that proline often is present in the looping regions of an alpha helix. And if it's in the middle of a alpha helix, it can cause problems given its structure, and this can cause misfolding. Finally, we can move to the tertiary structure, and this is a bit more involved than either the primary or secondary structures, in that it involves hydrogen bonds, the hydrophobic effect, as well as the disulfide bridge. Now, with the disulfide bridge, you have 2 cysteine amino acids somewhat near each other, and this results in a disulfide bond where 2 sulfur atoms can be bonded together, and this is another force driving that tertiary structure. So 2 beta plated sheets in this example are able to come together via this disulfide bridge. Also, you can have hydrogen bonding, as we see here, between the alpha helix and the beta plated sheet, driving tertiary structure. Also, you can have the hydrophobic effect, whereby non-polar amino acids will want to come together because polar will not want to go near non-polar, and so these will come together, and this can cause clumping if on the outsides you have more interactions with polar amino acids driving things inward. And this is how you get an interior structure of proteins. And this is exemplified in the quaternary structure, which also relies on the hydrophobic effect at a greater scale. And so you can see that the interior of 2 subunits, which is what makes up the quaternary structure, you have 2 subunits, 3 subunits, you can have 4, and like in hemoglobin. And then the hydrophobic effect will develop in the interior of that protein structure, and maybe polar amino acids will be moved outward. In addition to the hydrophobic effect, you can again have hydrogen bonds driving a lot of the connection between various different subunits. But you also can have ionic interactions between R groups. For example, here we can see that lysine and aspartic acid are positive and negative respectively, and an ionic interaction can form here, and that's a pretty strong interaction that will allow these two subunits to remain near each other, connected and bonded together. So those are the four major layers of protein structure that scientists have been able to model out and develop for us. Now we can take a look at a specific reaction that can take place within proteins. So let's say that this fictional protein, for example, is a protease, a serine protease, whose function it is to cleave peptide bonds in other proteins. So for example, when you eat food that has protein in it, it'll eventually reach the small intestine and can be digested by proteases, such as this example we are about to run through. And it is driven primarily by its active site, which consists of a catalytic triad of serine, histidine, and aspartic acid. So the serine amino acid is going to act as a nucleophile and attack the nucleus-loving carbonyl of the peptide bond. Of the polypeptide, it is going to break down, cleave, and digest. This causes an electron to move up to the oxygen of the carbonyl. Meanwhile, and simultaneously, histidine is taking the H of serine and stabilizing it within its structure, while aspartic acid is stabilizing histidine and its structure. So these are all stabilizing forces, allowing serine to act as a powerful nucleophile on the carbonyl within a polypeptide that it wants to digest. This allows for a tetrahedral intermediate. Tetrahedral intermediates are very common and very important. And so, with this intermediate, you can have the oxygen that had that electron move up, move back down. This will cleave the peptide bond specifically, such that it will move towards the histine that has the NH that it was stabilizing. And voila, you have the peptide bond cleaved. You now are left with the carbonyl and NH2. This will happen at each of the peptide bonds that come within range of our catalytic triad in the active site of our protease. And this digested protein will make for a much easier candidate to be absorbed by our cells and tissues within the small intestine, which we can then use for various different other metabolic processes that drive our bodily function. And so with that, I will stop there and see you guys in the next one. Take care.